Welcome to Parasitology. Hi, I'm Christopher. Parasitology is an amazing topic. But before we start learning parasite names, diseases, and life cycles, we need a simpler question. What exactly is a parasite? More importantly, we need to learn the language of parasitology. These terms are not just vocabulary words. They are tools that help us understand how parasites live, how they move, how they cause disease, and how we diagnose and prevent them. What is a parasite? A practical definition, adapted from Georgie's Parasitology for Veterinarians, is this. A parasite is an organism that lives on or in a host and benefits at the host's expense. In veterinary parasitology, we usually focus on several major groups: protozoa, helminths, and arthropods. In a broad biologic sense, some bacteria, viruses, and fungi can also be considered parasitic. But conventionally, we separate those into other disciplines. Don't worry, there are more than enough parasites for us to handle here. To think like a parasitologist, and honestly to think like a veterinarian, we need to speak the language of the parasite names. Parasite names follow the same binomial naming system used for other organisms. The genus name is capitalized and italicized. The species name is lowercase and italicized. For example, Toxocara canis is the dog roundworm. Once the genus has been introduced, it can be abbreviated to T. canis. These names matter because they appear in textbooks, journal articles, diagnostic reports, and product labels. They are not trivia. They are part of the language of veterinary medicine. Taxonomy is a way of organizing our thinking. It's not perfect, and parasite names sometimes change as our understanding of relationships changes. That can be frustrating, but taxonomy gives us a useful framework. It helps us organize parasites by shared evolution, anatomy, life cycles, hosts, and clinical behavior. So the goal is not just about memorizing names. The goal is to use names and groups to make better predictions about biology, disease, diagnosis, and control. Next, we need to talk about symbiosis. Symbiosis simply means that two organisms live in close association. That association can take several forms. In mutualism, both organisms benefit. In commensalism, one organism benefits while the other is not really meaningfully helped or harmed. In parasitism, one organism benefits at the expense of the other. That last category is our focus. But here's the important clinical point. Finding a parasite does not automatically prove that the parasite is causing the patient's problem. Parasites may cause obvious disease, subtle disease, or no clinically apparent diseases at all. The distinction between infection or infestation and disease will matter throughout parasitology. Parasites can also be described by where they live. Ectoparasites live on the surface of the host or within the skin. Fleas, lice, ticks, and many mites are examples. Endoparasites live deeper inside the host, such as the intestine, liver, lungs, blood, or other tissues. This distinction also affects our language. Ectoparasites are often said to cause infestations, while endoparasites are often said to cause infections. That's not just a word choice. Location shapes clinical signs, diagnostic choices, treatment decisions, and prevention strategies. Some parasites are obligate parasites, meaning that they require a host to complete their life cycle. Others are facultative or opportunistic. These organisms live independently but may become parasitic when the opportunity arises. Naegleria fowleri is one example. It is often called the brain-eating amoeba in public-facing language, but taxonomically, it's better described as an amoeba-like heteroloboscean protist. It normally lives freely in the warm fresh water and soil, but under the wrong circumstances, it can enter through the nasal passages and cause primary amoebic meningoencephalitis. The broader lesson is not that every parasite-host relationship looks the same. Some parasites are committed to life in a host, while others become parasitic only under specific circumstances. Parasites vary in host specificity. Some are highly host specific and prefer one host species or a narrow group of related hosts. Many lice are classic examples. Others are less host specific and can infect a broader range of animals. Host specificity matters because it helps us predict risk, transmission, disease severity, and zoonotic potential. When we ask, can this parasite infect this patient? We are asking about host range. Now let's talk about one of the most important ideas in parasitology: host roles. A definitive host is a host in which the parasite reaches sexual maturity or undergoes sexual reproduction. An intermediate host is a required host in which a parasite undergoes development, asexual reproduction, or larval growth, but does not reach sexual maturity. A paratenic host is a transport host. The parasite remains alive and infective, but does not undergo required development. An aberrant host is an unusual or accidental host. In these hosts, the parasite is not where it's supposed to be. Development may become incomplete, migration may become abnormal, and disease may be more severe. Not every parasite uses every host category. Some have direct life cycles with only one host. Others have complex indirect life cycles involving multiple hosts. The terms are not meant to trap you, they're meant to give you a map. Disease severity often depends on the parasite's life stage, tissue location, and the host's role in the life cycle. If we anthropomorphize this a little, many parasite life cycles seem to have a method to their madness. Almost as if the parasite is working its way through an escape room, trying to reach the definitive host where it can mature and reproduce. Of course, the parasite is not thinking or planning. Natural selection is the escape room designer. In some life cycles, disease in the intermediate host may increase the chance that the parasite reaches the next host. Sometimes that means multiplication within the intermediate host, and sometimes it means tissue damage, weakness, abnormal behavior, or increased vulnerability to predation. A useful example is Tinea multiceps. Adult tapeworms live in the intestines of the dog, the definitive host, and often cause little obvious disease. In sheep, the normal intermediate host, the larval cyanurus stage can develop in the brain and cause GID with circling, ataxia, blindness, recumbency, or death. That does not mean that the sheep is simply a poorly adapted host. The sheep is part of the life cycle. But the parasite stage and location can cause severe disease and neurologic impairment that can increase the chance that a canid eats or scavenges the sheep. So the takeaway is this: host role matters, but so do parasite stage and location. Parasites follow patterns, and those patterns can look sneaky, but they also love exceptions. Let's define vectors. A vector is an organism that transmits a pathogen or parasite from one host to another. A mechanical vector transmits the agent without required development or multiplication inside the vector. The vector acts as a contaminated carrier. For example, ticks and other arthropods may mechanically vector swinepox virus between pigs. A biologic vector is different. In a biologic vector, the agent undergoes required development, multiplication, or both. Mosquito transmission of malaria is a classic example. The malaria parasite develops and multiplies inside the mosquito before transmission to another host. Phoresis is related but different. Phoresis means that one organism uses another for transport. the transported organism is not necessarily feeding on or infecting the carrier. A simple teaching example is a housefly landing on food and passively carrying bacteria onto a pizza. The key idea is movement. Vectors transmit, Phoresis transports. And in parasitology, movement between hosts is often half the battle. Reservoir hosts are another important concept. A reservoir host is an animal population that helps maintain a parasite or pathogen in nature. Reservoir hosts may or may not be the main species we care about clinically, but they keep the cycle going. Geography and population patterns also matter. When a parasite consistently is present in a region or population, we often call it endemic. For animal diseases, the more precise term is enzootic. When disease occurs at unusually high levels in an animal population, we call it hyper-enzootic. Prevalence and incidence are also different. Prevalence is the percentage of infected individuals in a population at a given time, while incidence is the rate of new infections occurring in a population over a defined period. Those two ideas are not the same. Prevalence helps explain burden, incidence helps explain risk, and both help us understand the parasite better. Finally, we need to talk about zoonotic language. In common use, a zoonosis is an infection or disease transmitted from animals to people. More precisely, animal to human direction is called anthropozoonosis. In other words, zoonosis is the common shorthand, while anthropozoonosis is the more specific directional term. Other directional terms also exist. Zooanthroponosis, sometimes called reverse zoonosis, refers to disease in animals acquired from humans. Amphizoonosis refers to infections that can move interchangeably between humans and other animals. Anthroponosis refers to infections maintained primarily among humans. A simple way to remember these terms is to look at the word parts. Anthro refers to humans, zoon refers to animals. In these directional terms, the first part generally tells us who has the disease, and the second part points towards the source. You do not need to use every term in daily conversation, but the language helps clarify direction of transmission. There are even more precise terms for transmission between wildlife and domestic animals. Zootherionosis refers to disease in domestic animals acquired from wildlife. Zootithocinosis refers to disease moving from one domestic animal species to another. Theriotithocinosis refers to disease in wildlife acquired from domestic animals. The goal is to understand the pattern. Parasites move through hosts, environments, vectors, food webs, and ecosystems. If we understand the life cycle, we predict disease. If we can predict the disease, we can choose better diagnostics, treatment, prevention, and public health recommendations. This is the language of parasitology. As we move through parasitology, keep asking those same core questions. Where does the parasite live? How does it enter the host? How does it move from host or environment? What damage does it cause? What stage can we detect? And how does it reach the next host? Those questions turn parasite terminology into clinical logic. That is where parasitology becomes useful, memorable, and genuinely fascinating. I'm Christopher Lee. Thank you for watching. And what you know today helps protect everyone tomorrow.